Assalamu alaikum everyone. In this video lecture, we will be discussing paper 1 of AS level biology 9700. The paper is from the year 23. The paper is of February March and the variant is 2. Let's start. MCQ number 1. The photomicrograph shows cells from a human blood smear. So we can see that this is a human blood picture of blood smear containing red blood cells and the neutrophil. Which calculation shows a correct method to calculate the actual diameter of the neutrophil shown in photomicrograph in micrometers? So guys, we have to select a correct option for the correct calculation of the actual diameter of this cell, which is the neutrophil. So for that you use the formula magnification is equals to image length divided by actual length, and you need to rearrange this formula because you want to find actual length. So when we rearrange the formula, this will be actual length is equals to image length divided by magnification. So you need to put image length and magnification in this formula to find the actual length. Now the image length will be the length of line PQ, and you all know that we always measure the length in millimeters, right? When we measure the length using the scale, we measure the length in millimeters. We always measure in millimeters, right? So image length that we will measure, that will be in the millimeters, and we need to convert it into micrometers because in this formula we need to put every value in micrometers. So simply the working will be length of line PQ in millimeters, right? And what you will do, you need to convert this into micrometers because you have to put this image length into the formula. So you need to convert it into micrometers by multiplying by 1000, and then you need to divide it by the magnification of the photomicrograph. So this the answer of this MCQ will be A. Let's move on to MCQ number 2. A student calibrated an eyepiece graticule using a stage micrometer. Each division of the stage micrometer was 0.1 millimeter. With into 10 magnification objective lens, 10 eyepiece graticule units matched 10 divisions on the stage micrometer. So guys, basically they are saying that at the magnification of into 10 eyepiece graticule units, 1 2 3 4 5 6 7 8 9 10. So these are 10 divisions from 0 till 10, right? 1 2 3 4 5 6 7 8 9 10. So 10 divisions of eyepiece graticule matched 10 divisions of stage scale. So we can draw the stage scale divisions with green, right? So one stage scale division was equal to the one eyepiece graticule unit. That's because 10 eyepiece graticule units were matching 10 stage scale divisions. All right. The same microscope was used with a into 40 instead of into 10 magnification objective lens to measure the diameter of an alveolus. The diameter of the alveolus was found to be 96 eyepiece graticule units. The eyepiece length was not changed. What is the best estimate for the diameter of the alveolus? So guys, what happened is that they used the same microscope, but this time with an into 40 objective lens. So always remember this point, that whenever the magnification of the objective lens is changed, the scale that is magnified is the stage micrometer scale, right? When you change the magnification of the objective lens, the stage micrometer is magnified. Over here they have mentioned that the eyepiece lens was not changed. This means that, for example, if now we are dealing with into 40 magnification, so the eyepiece scale would remain the same. However, the stage micrometer will now be magnified 4 times, right? It will be magnified 4 times because the magnification is now into 40, right? So what will be the case now? Now almost we can say that one stage micrometer division will now be equal to 4 eyepiece graticule units. Do you get this point or no? Initially, what happened that 10 graticule units was equal to 10 stage scale divisions. Now what happened is that the magnification has changed to into 40. So what has happened is that the stage micrometer has been enlarged. So one stage scale division will now occupy more graticule units, right? So initially at into 10, one graticule unit was equal to one stage scale division. So now what will happen is that at 40, at into 40, what will happen is that more graticule units will be covered by one stage scale division because the stage scale division has been enlarged. And how many times has it enlarged? 4 times. So the graticule units will be multiplied by 4. And why the stage micrometer scale or division has enlarged 4 times? Because the magnification has changed from into 10 to into 40. So 4 graticule units is equals to 1 stage scale division. So we can also say that 4 graticule units is equals to 0.01 millimeter, because 1 stage scale division is equal to what? Is equal to 0.1 millimeter. This means that 1 graticule unit will be equal to 0.01 millimeter divided by 4. We can also convert this into micrometer. 1 graticule unit is equals to 0.01 into 1000, because we want to convert it into micrometer, divided by 4. So this will be, when you multiply 1000 into 0.01, this will be 10 divided by 4 is equals to 2.5 micrometers. So now 1 graticule unit is equals to 2.5 micrometers. The number of graticule units measured, or we can say that the diameter of the alveolus is equals to 96 graticule units. So 1 graticule unit is equal to 2.5 micrometers. So 96 graticule units will be equal to x. If we cross multiply, the x will be 96 into 2.5, that is 240 micrometer. So alveolus has a diameter of 240 micrometers. We are done with MCQ number 2. Let's move on to MCQ number 3. Which statements are correct for chloroplasts and also for mitochondria? They contain ATS ribosomes. No guys, chloroplasts and mitochondria, they contain 70S ribosomes. They can transcribe their circular DNA. This is true. Both chloroplast and mitochondria, they have circular DNA, and the transcription of circular DNA can occur in these organelles, right? Because these organelles contain RNA polymerase enzymes, and they can transcribe their circular DNA. They can translate messenger RNA. Yes, both chloroplast and mitochondria, they can translate messenger RNA, or they can carry out translation. That's because they have 70S ribosomes. They're enclosed by double membranes. Yes, both chloroplast and mitochondria are enclosed by double membranes. The answer is C, 2, 3 and 4. Let's move on to MCQ number 4. The electron micrograph shows some cells from a root. Which cell structure is not usually found in the cells from a root? So guys, if we look at this diagram, A is the nucleolus. A is the nucleolus. D is the nucleus. right? Because this entire structure is the nucleus, and in the center of the nucleus is the dark structure that is nucleolus. B is the chloroplast. If you look at B, so B is the chloroplast, and C is the cell wall. So they're saying that which cell structure is not usually found in the cells of the root. So you need to know that chloroplasts are not usually found in the cells of the root, because roots are below the ground where the light does not reach. So photosynthesis cannot occur and there is no need of any chloroplast. So chloroplasts are not usually found in the root. Let's move on to MCQ number 5. Dialysis whisking tubing is an artificial partially permeable membrane with pore sizes of approximately 2.5 nanometers. Glucose molecules have a diameter of about 1.5 nanometers and can pass through the pores in the membrane. What else can pass through the pores? So guys, they're saying that there's a artificial partially permeable membrane, that is whisking tubing or dialysis tubing, and it has pores with sizes of approximately 2.5 nanometers, and glucose molecule has a diameter of 1.5 nanometer, so it can pass through the hole. We need to select the options that can also pass through these pores. So bacteria cannot pass through these pores because the size of the bacteria is 0.5 to 5 micrometers. 0.5 to 5 micrometers. If we convert this into nanometers, this will be 500 nanometers to 5000 nanometers, and this cannot pass through the pore, which is 2.5 nanometer in diameter. Hemoglobin cannot pass as well. That's because hemoglobin is a large protein molecule. It is made up of many amino acids. So if glucose has a diameter of 1.5 nanometers, so amino acid will have somewhat the same diameter, because glucose and amino acids usually have the same size. But you need to know that hemoglobin is a very large protein molecule made up of many amino acids, so obviously it has a size much greater than 2.5 nanometers. So hemoglobin cannot pass. Ribosomes cannot pass because they have a size of 25 nanometers, greater than 2.5 nanometers, and fructose can, however, pass because fructose is a monosaccharide, is a hexose just like glucose, and they almost have the same size. So only fructose can pass. The answer is D, 4 only. MCQ number 6. What is present in all viruses? So guys, viruses, you need to know that viruses contain either DNA or RNA. They cannot contain both, either DNA or RNA, right? Either DNA or RNA. So a virus could be a DNA virus or it could be an RNA virus. They cannot contain both. So if we talk about what is present in all viruses, ribose is not present in all viruses. It is only present in RNA viruses, right? Viruses which contain RNA will contain ribose. So ribose is not present in all the viruses. It is only present in RNA viruses. If we talk about deoxyribose, it's not present in all the viruses. It's only present in DNA viruses, right? If we talk about adenine, so adenine is present in all the viruses, because if the virus is a DNA virus, it will contain adenine. If a virus is RNA virus, it will contain adenine. That's because adenine is present in both DNA and RNA, right? So all the viruses contain adenine base. Thymine is not present in all the viruses. It is only present in DNA viruses, because thymine is only found in DNA, right? So the answer is C. Let's move on to MCQ number 7. To estimate the concentration of glucose in an unknown solution, equal volumes of range of known concentrations of glucose were each mixed with the same excess volume of Benedict's solution. After mixing, the solutions were placed in a thermostatically controlled water bath at 90 degrees Celsius for 3 minutes. The unknown solution was then treated in the same way, and the colors of the known and the unknown solution are compared. What is the independent variable in this procedure? So guys, over here, what they are doing is that they have to estimate the concentration of glucose in an unknown solution. So they are basically picking up a range of glucose solutions with different concentrations, right? And for example, they will pick up 5 different solutions with different concentrations of glucose, and then they will carry out Benedict's test, and they will find out the colors, and then they will repeat the Benedict's test on the unknown solution, and then they will compare the colors to find out the concentration of glucose in the unknown solution. So they are asking us about the independent variable in this procedure. Always remember this point, that independent variable is a variable that we change ourselves directly, and independent variable is the variable which does not depend on the other variables, right? So the concentration of glucose over here is the independent variable, because this is something that we are changing directly, right? And on the concentration of glucose, the color of the Benedict solution, or the final color of the solution, depends. So this is the dependent variable, right? Temperature of the water bath over here is a controlled variable. It's a standardized variable. We are maintaining temperature at 90 degree. We are not changing it. And volume of glucose solution over here is also a controlled variable. So what is being changed in the investigation is the independent variable. The answer is A, concentration of glucose. MCQ number 8. Which statements are correct for amylose and also for amylopectin? So they both are carbohydrate molecules. This is true. They are formed by condensation reactions. This is true as well. Amylose and amylopectin, they are the polymers, and they are made up of alpha glucose. Alpha glucose molecules are joined together by condensation reactions. They are linear molecules. No guys, they are not. Amylose is helical, not linear, and amylopectin is a branched molecule. So these molecules are non-linear, right? And they contain alpha 1,4 glycosidic bonds. Yes, both of them contain alpha 1,4 glycosidic bonds. Additionally, amylopectin contains alpha 1,6 glycosidic bonds. So the answer is 1, 2 and 4. The answer is B. Let's move on to MCQ number 9. Which statement is correct? Which statement is a correct comparison between saturated triglycerides? Triglycerides are fats, guys. So which statement is a correct comparison between saturated triglyceride molecules or saturated fats and unsaturated triglyceride molecules or unsaturated fats of approximately the same molecular masses? So guys, they are saying that which statement is the correct comparison between saturated fats and unsaturated fats of approximately the same molecular mass. So unsaturated triglycerides or fats have more double bonds. This is true. Unsaturated triglycerides have more double bonds as compared to the saturated fats or triglycerides, and fewer hydrogen atoms than saturated triglycerides. This is true, guys. This is the answer. You need to know that unsaturated fats or unsaturated triglycerides, they have more double bonds, and if there are more double bonds, it means there are less hydrogens, because this is the rule. Greater the number of double bonds, lesser will be the hydrogens. Let's move on to MCQ number 10. The diagram represents a molecule from a cell surface membrane. So guys, this is a molecule from the cell surface membrane, which means that this molecule is a phospholipid, right? And what is A? In this molecule, A is the hydrophobic tail, right? Or the fatty acid tail. If we talk about D is the phosphate group, right? B are the hydrophobic tails, right? And C is glycerol plus phosphate, right? Or we can say C is the hydrophilic head. C is the hydrophilic head, right? Which description of one of the labels is correct? Fatty acid at the hydrophilic end of the molecule. Guys, A can be labeled as fatty acid, but we can label A as fatty acid, but this is not at the hydrophilic end of the molecule. This is at the hydrophobic end of the molecule. This is the hydrophobic end. Hydrophobic end of the triglyceride molecule. This is not a triglyceride molecule, guys. Hydrophobic end of the glycerol molecule. There is no hydrophobic end of the glycerol molecule, right? Phosphate group at the hydrophilic end of the molecule. This is true. This is the phosphate group at the hydrophilic end of the molecule. So answer is D. C says hydrophobic end at the glycerol molecule. So guys, glycerol is usually hydrophilic, not hydrophobic. And we can say that this is the end. We can usually say that this is the hydrophobic end of the glycerol, but it's not labeled over here, so C is wrong.